Neurons are made up of three parts: a cell body, multiple dendrites, and a single axon. The dendrites receive information, while the axon transfers information to other neurons. Let's start with the cell body. The cell body, which is sometimes referred to as the perikaryon or soma, houses the nucleus and other organelles within its cytoplasmic membrane. In this image, we can see the cell body highlighted in green. To identify them in histological slides, we can look for the structure that is dilated in shape, so larger and usually rounder than the projections. Here's another example of a cell body in this image. Another way to identify the cell body is by identifying the nucleus within it. This is the structure we can now see highlighted in green on the left. In this stain, it appears darker than other structures in the cell body. It will be spherical and the largest structure within the cell body. The nuclei are unusually large in neurons. As in all cells, the nucleus houses DNA. On the left, the many small granular structures we can see highlighted in the cell body of this neuron are called Nissl bodies. Nissl bodies are granular collections of rough endoplasmic reticulum and free ribosomes and can be found throughout the cell body. Since these structures contribute to protein synthesis, this shows that neurons have a high synthetic activity. Depending on the activity of the neuron, the number of Nissl bodies will vary. The soma, or cell body, has numerous cytoplasmic projections branching from its surface. We can see two in this image highlighted in green on the right. These are dendrites. Dendrites form connections with other neurons in order to receive information, so they're conducting impulses towards the cell body. There are smaller projections called dendritic spines that branch out from each of the dendrites. Unfortunately, we can't see these on the histological slide. Dendritic spines are the parts of the dendrite that form synapses with other cells. A synapse is where the communication occurs between two cells. Synapses can occur not only between two neurons, but also between a neuron and a muscle fiber. If a synapse is occurring within another neuron, the dendritic spines of one neuron are forming that synapse with the axon of another neuron. The axon is a single projection from the cell body that carries information from the cell body to another cell. To differentiate axons from dendrites, you can look for the following things. 1. Axons are usually much longer than dendrites. Their length can vary, but axons can be over a meter long. 2. Axons run a straighter course than dendrites. Dendrites will usually bend and twist to meet an axon, not the other way round. 3. Branching of axons is limited to the distal end, whereas dendrites might branch earlier. And 4. Finally, where axons originate from the cell body is quite distinct. The axon originates from the cell body via the axon hillock. This is the pyramid-shaped region that is highlighted in green on the image on the right. In this new image, we're looking at a cross-section of a peripheral nerve. Within the nerve are many neurons, some of which are shown in this zoomed-in section. When looking at this image, we have to imagine that the nerve is travelling towards us, so the structures highlighted in green are the cut ends of axons travelling towards us through an insulating sheath. This insulating sheath surrounding the axons is what we can now see highlighted in green. This sheath is known as myelin. Its main purpose is to increase the speed of the impulses travelling along the neuron. When we take the green highlight away, we can see that the myelin is in fact a white space. That is because during the staining process, the alcohol involved removes the myelin, and an empty space is what's left. Not all axons are myelinated, however. This new image on the right shows us what unmyelinated axons look like, again from a cross-section. They're much thinner since they don't have several layers of myelin wrapped around the axons. We can see the axons of the unmyelinated neurons within the green circles.